You dropped the term erythropoietin.
Yes.
And that is a ligand for these receptors that we're using, the cytokine receptors.
Yeah.
Could you please elaborate how is this molecule, erythropoietin, important in the brain? As long as I know, it's produced by the kidneys and liver, and it stimulates red cell production. But what is the role in the brain of this molecule?
So the name erythropoietin is coined because the stimulation of red blood cell production was the first function described for this molecule. And as you correctly mentioned, it's typically released by the kidney in situations when there is a lack of oxygen in the tissue, and the more erythropoietin is built, then more red blood cells will be produced, and these red blood cells carry the oxygen in the organism, and therefore we have a better oxygen distribution. And the erythropoietin in these blood production system basically prevents the degeneration, the cell death of the developing red blood cells. So developing red blood cells without the EPO signal will undergo self-destruction, which we call apoptosis. It's a regulated program that kills cells, and therefore only if there is an erythropoietin available and activates the receptors, these cells will be mature cells that can really transport the oxygen in the organism later. And basically later people have found that erythropoietin is also produced and released in other tissues, including the nervous system. And the stimuli that lead to the production and release of erythropoietin in the nervous system are very similar. So there's also hypoxia, there is toxins, inflammation, things like this, mechanical injury, but it's also high neuronal activity. So also in terms good activity related to social enrichments and all these things. So whenever there is local high activity in the brain, it seems like erythropoietin is produced and acts on neighboring cells that express a receptor for erythropoietin. And the typical erythropoietin receptor that we have on our red blood cell, that's a molecule which is very characteristic, and it's also present in the nervous system. But we as humans and other mammals also produce a variant of the erythropoietin molecule, which cannot activate this receptor, this specific receptor, but can still mediate protective effects on nerve cells. So in order to achieve this, there must be another receptor to distinguish between these two functions. And this was basically the idea why we started with the work on insects, because insects don't produce erythropoietin. Only with the evolution of the vertebrates, all these signaling substances evolved. So insects don't have this, but still the human erythropoietin can save the insect neurons by activating the same pathways that are activated, for example, in our brains.